I'm Rick Lifton, President of the Rockefeller University, and I'm delighted to welcome you to this webinar for our benefactors and friends, sponsored by the Rockefeller University Council and the Committee on Trust and Estate Gift Plans. I'm pleased to have my colleague, Dr. Barry Coller, joining us to report on his amazing translational research in this program entitled Racing Against Time, a New Approach to Battling Heart Attack. This is the season when we usually take pleasure in welcoming you to the Rockefeller campus because in addition to being able to share the transformative discoveries happening in our laboratories on a daily basis, we're able to share the wonderful transformation that spring brings to our spectacular gardens. This year, of course, our program is taking place in the virtual setting of a Zoom webinar because of the need for continued social distancing. But just as the blooming of Rockefeller's azaleas reminds us that long winters do eventually come to an end, so too are we seeing the proverbial light at the end of the tunnel in this pandemic with the vaccination of millions of people around the globe, including, as of this afternoon, more than 120 million people in the United States. Over the last year, we've seen unprecedented efforts by scientists working to understand the SARS-CoV-2 virus and to develop vaccines, antibody treatments, and other therapies and tests for COVID-19. The level of collaboration among academicians and between academicians and industry has truly been unprecedented. This has resulted in progress at an astounding pace, bringing us closer to being able to visit loved ones and return to the business and activities that have been curtailed by the virus. I want to thank the members of the Council and the Committee on Trust and Estate Gift Plans for directing generous philanthropic support to our COVID-19 efforts, as well as for their ongoing contributions, which continue to sustain our research and educational activities at Rockefeller. To all of our donors, we thank you for your wholehearted and magnanimous support over the tremendously challenging past year. Philanthropy has always played a critical role at Rockefeller, where support of the most gifted and creative scientists has led to an exceptional record of accomplishment. Over the years, 26 of our faculty at the university have received a Nobel Prize, including 3 in just the last 10 years, a number matched by no other institution in the world. This includes the 2020 Nobel Prize presented in December to virologist Charles Rice, who received the prize in medicine for his seminal contribution that led to the cure for hepatitis C, a global scourge causing death by liver failure. In the last decade, Rockefeller University has created a robust translational research ecosystem through the establishment of our Robertson Therapeutic Development Fund and other vital programs, such as our new Women in Science Entrepreneurship Fund. These initiatives are ever more important as we witness the increasing convergence of basic science and clinical medicine. Our translational research programs provide Rockefeller scientists with many new opportunities to develop therapeutic applications of their discoveries. A leader in these translational research efforts is today's speaker, Dr. Barry Coller. A specialist in vascular biology and blood disorders, Barry joined Rockefeller in 2001 as Vice President for Medical Affairs, David Rockefeller Professor, and Physician-in-Chief of the Rockefeller University Hospital. He came to the university from Mount Sinai School of Medicine, where he chaired the hospital's medical service and was chair of the medical school's Department of Medicine. He oversees the training and performance of clinical research at the Rockefeller Hospital and serves as director of the university's Center for Clinical and Translational Science and of our Maurice R. and Corinne P. Greenberg Center for the Study of Inflammation, Microbiome, and Metabolism. In addition, Barry heads the Alan and Frances Adler Laboratory of Blood and Vascular Biology, which investigates platelet physiology and thrombosis, the formation of blood clots. Platelets are small blood cells that stop people from bleeding by sticking to damaged blood vessels, bringing in more platelets to plug the holes and activating the clotting system that stabilizes the clot. Unfortunately, platelets can also cause blood clots in one or more of the arteries that supply blood to the heart, the coronary arteries. The blood clot is the most common cause of heart attacks because it abruptly stops blood from reaching part of the heart, and as a result, the cells in that part of the heart die. The ultimate goal of Barry's research is to develop new therapies for thrombosis-related diseases, including heart attack and stroke, which, despite great progress over the last 50 years, remains the leading cause of death worldwide, with cardiovascular disease causing 18 million deaths per year. Moreover, you may have seen the recent news about a very rare complication of the AstraZeneca and now perhaps the Johnson & Johnson vaccines that likely cause blood clots and low platelet levels after vaccination in very rare instances. These appear to result from abnormal activation of platelet activities. Barry's past research on platelets led to the development of the drug Abciximab, known commercially as RioPro. Abciximab was approved by the FDA in 1994 and since then has been used to reduce complications of coronary artery angioplasty and stent placement in millions of patients worldwide. In recent years, Barry has continued to seek ways to improve the outcomes for heart attack patients. Despite the high efficacy of clot-busting drugs and stents in the treatment of heart attacks, it has been well known that up to 30% of deaths from heart attack occur before patients ever get to the hospital, making the creation of an effective and safe antiplatelet drug to be given during the critical pre-hospital period a goal of great importance. Today, he will share with us the path to the creation of a new small molecule known as RUC4 with the generic name zalunfiban that prevents blood platelets from sticking together and is crucially designed to be administered in an ambulance or even at home via a simple injection device. The development of RUC4 is a superb example of the convergence of great basic science and clinical medicine at Rockefeller. This work has relied on the resources of the university's shared technology centers, the hospital, and the Center for Translational Sciences. Vital support has also been provided by an Early Clinical Development Fund award from our Robertson Therapeutic Development Fund. Barry has been a leader in advancing translational research on our campus and beyond. He's also been active in shaping national policy governing biomedical research. In 2017, he served on the National Academy of Sciences Human Gene Editing Committee, which addressed issues related to use of the new CRISPR technology in patients. Barry is a member of both the National Academy of Sciences and the National Academy of Medicine. As we listen to Barry tell the exciting story of his new drug, you're welcome to submit questions at any time during the talk by typing them into the Q&A function at the bottom of your screen. At the end of the presentation, Barry and I will try to answer as many questions as possible. And Barry, it's now my great pleasure to turn the screen over to you.
Thank you, Rick, for that very kind and generous introduction. And I'm thrilled to have the opportunity to speak to you today about my research and about the challenges we face still with regard to heart disease. I think we've all become amateur epidemiologists this year because of COVID. And you can see on this slide that the estimate is that someone has a heart attack in the US every 40 seconds, and that's about 800,000 Americans having a heart attack each year. And there are approximately 365,000 deaths each year from coronary heart disease, which is primarily due to heart attacks. And this means that every 16 or 17 months, we have as many people dying from this disorder as have died from COVID-19 in the last year. As Rick indicated, the key to understanding the heart attack is to see that the muscle dies if the blood vessel becomes occluded. And the blood vessel becomes occluded with an underlying atherosclerotic plaque, but then almost always the final trigger is a clot that forms on top of that plaque. And just to show you what that plaque looks like in a real artery, all of this should be white. This should be an all open artery, but you can see this gruel-like material that is the atherosclerotic plaque. Work by Joe Goldstein, a trustee of the university, led to the development of statins, which have had a profound impact in reducing atherosclerosis and therefore have a profound impact on the reduction in the number of heart attacks we've had in the United States over the last several decades. The thing that causes the heart attack, though, is usually this acute clot on top of that chronic atherosclerotic process. And so both processes are very important. And to just emphasize what Rick indicated, ischemic just means that the heart is not getting enough blood, which is by the mechanism I just mentioned. And you can see that this is the #1 cause of death globally in 2016. And stroke is #2. And stroke, a portion of the stroke is due to a very similar mechanism. Another portion is due to bleeding, to hemorrhage. And then you can see the other causes of disease are really very interesting, but you can see that they are, in fact, relatively less common, and the importance of ischemic heart disease. And in the newly industrializing countries, we're seeing a very profound increase in heart disease that really mimics what happened in the United States from the turn of the 19th century through about 1960, 1970, which is when we reached our peak of heart disease. And here you can see both in urban and rural areas in China, this really dramatic increase in the number of acute myocardial infarctions. And acute myocardial infarction is, in fact, the fancy name for a heart attack. But you can see how dramatic this is. And if we take a look at the 1.4 billion people, you can see that this number of about 70 myocardial infarctions per 100,000 people turns out to be about 1 million deaths per year. And this is really the key slide for today. And what it says is that if you close a coronary artery, either in a human or in an animal, that would ordinarily lead to death, if you open it immediately, no one dies. You get 100% mortality reduction. But then over a period of just a couple of hours, the ability to salvage, to not die as a result of that occlusion goes down this very, very steep slope. And so time is absolutely crucial in improving our treatment of heart disease. In our country, most patients will be immediately transported to a center that has the ability to perform the procedure that's illustrated on this slide, which is called a percutaneous, meaning you go through the skin, coronary, meaning the coronary artery, and intervention. And the intervention consists of putting in a balloon that's deflated with a stent around it. And the balloon then gets inflated in the area that's obstructed, and the stent then gets deployed. And when that happens, the balloon is deflated and the stent stays in place to keep the blood flow open. But whenever you do this, you're going to damage the atherosclerotic plaque, and that means platelets will come in. And so all of this technology really required simultaneous development of better antiplatelet therapy. So the platelets really were discovered much after the other cells in the blood, so the white blood cells and the red blood cells, but the platelets are much smaller, and they're actually not even full cells. They don't have a nucleus, they're just fragments of cells. And they circulate in the blood in an inactive form, and the inactive form sort of looks like pita shaped. But if you begin to activate them, and I'll go into a slide that shows you that there are many different things that can activate platelets, they immediately start to change shape in a rather dramatic way. And then over here, you can see that they start sticking one to another. And this sticking process is the basis for stopping bleeding, but it's also the basis for developing the clots that can close off an artery. And on this slide, I want to just focus on a couple of things. This is the inside of the blood vessel, and this is the lining of the blood vessel that has the term endothelium. And ordinarily, the endothelium actually secretes things that keeps platelets inactive. But when you damage the endothelium, like when you cut yourself, or when the atherosclerotic plaque gets damaged, you will begin to expose a variety of proteins under that endothelium. And as a result, the platelet will begin to stick, much like Velcro, because platelets have a series of receptors that interact and will form interactions with the different proteins that are exposed over here. And then the next step that occurs is that the activators get out of the platelet, and also signals come from these receptors that activate this receptor. And this is a receptor we're going to talk about primarily today. And it goes by two names, alpha-2b beta-3 and also GP2b3a for historical purposes. When it gets activated, it changes its shape. You see it sort of bent over here, and I show it extended over here. And then it can do something really interesting. There's a lot of protein floating around in plasma, and two of those proteins, fibrinogen and another protein called von Willebrand factor, are very big. And in fact, fibrinogen has two ends that look just alike. And as a result, you have the ability for the platelets to actually clump together because they're bridged by these large molecules that can span the alpha-2b beta-3 receptors on the two different proteins. I just want to emphasize here, we're talking about heart attacks today, but many of you, I'm sure, have read about the fact that in COVID-19, there are many cases of thrombi forming in the lung, and they form exactly the same way. But in that case, the damage to the endothelium is occurring through viral infection. Although whether or not the endothelium itself is infected by the virus is still a matter of investigation. But there is no question that the endothelium is damaged, and that is what leads to those thrombi in the lungs that make a very important contribution to the problems in patients' ability to breathe. And I just want to show you what happens when you damage the endothelium. This is the carotid artery in the neck of a mouse, and the size of this is about the size of a narrowed coronary artery that has atherosclerosis in it. And you can see within a couple of minutes, all of this white stuff is just wall-to-wall platelets extending out from the damaged area. And you'll see that within 6.5 minutes, you can actually see that there'll be no more blood flow in this artery. And basically, this is the type of mechanism that is responsible for a heart attack. We've studied this receptor for many years and worked with groups that made mice that lacked the receptor. And if we do the same experiment in that type of mouse, you can see at 5 minutes, there really is just nice flow in this artery. And if we wait over here to 20 minutes, we're still seeing the same thing, even though the endothelium is damaged, because the blood vessels the platelets can't recruit additional platelets. Now, I know this slide is complicated, but let me walk you through it because it really is not as bad as it looks. So this is one platelet over here, and this is another. And here we're showing the platelets are connected by the fibrinogen molecule by bridging the alpha-2b beta-3. And I started by telling you that there are a lot of different things that can activate the platelet. And this is among the list of things that can activate the platelet. And the way they do it is by interacting with receptors that are on the platelet. And the key thing, though, is all of these different receptors, their signaling all leads to the same final common point, which is getting that alpha-2b beta-3 receptor to undergo that conformational change that allows it to bind the fibrinogen and cause the linking. And that's really the key point here. On here, we have all of the now available antiplatelet drugs. Aspirin is powerful in preventing the conversion of arachidonic acid into thromboxane, which is one of the things that can activate the platelet. Thrombin itself can activate the platelet, and anticoagulants can work against that. Vorapaxar is a relatively recent recent antiplatelet agent that blocks one of the receptors that thrombin interacts with. There are a series of different drugs that have been developed that block one of the ADP receptors. ADP is one of the more important activators of platelets. And then, as Rick mentioned, Abciximab was really developed from a monoclonal antibody that was prepared in my laboratory quite a number of years ago. And that antibody was called 73, and then it was reworked with the scientists at Centocor and developed into the drug Abciximab. The most important first for Abciximab was that it was the first antiplatelet agent that was developed in a way that I'll call is rational, meaning it was based on understanding the way platelets work, as opposed to doing screens or just the serendipity, which is the way people recognized that aspirin was an antiplatelet agent. And 4 years later, these two other drugs, which are small molecules, Abciximab is a derivative of the monoclonal antibody, but these two were then approved and bind to the same receptor. We can measure the way platelets clump together by having them suspended in a cuvette, just a glass cuvette that you can shine light through and see how much light gets through. If they start to aggregate, more light gets through because the amount of kicking back of the light is more a function of the number of particles than the size of these small aggregates. And so over here, if we add arachidonic acid, as I mentioned, that's going to get converted into thromboxane, and that's then going to cause the platelets to aggregate. And when they aggregate, more light gets through, and that's really what you're seeing here. If we take the platelets and first treat them with aspirin, they're not going to be able to aggregate because the arachidonic acid will not get converted into the thromboxane because the aspirin blocks that. So you can see that that's what aspirin can do. But if you use ADP, another activator, you can get the platelets to aggregate whether they're treated with aspirin or not. But if you put an antibody that will block where the fibrinogen ordinarily binds to alpha-2b beta-3, you won't get activation, you won't be able to get any aggregation with ADP or with arachidonic acid or any of those activators. And the drugs that I mentioned, the three drugs that work against alpha-2b beta-3, were really sort of models of clinical trials because there were over 40 clinical trials with over 31,000 patients. And as you can see here, there was about a 30% decreased likelihood of people having, of dying from having a heart attack at 30 days after the event, or a combination of either having a heart attack or dying was also reduced by about the same amount. There was an increase in bleeding. Fortunately, some of this was technical, and they changed the way they do the procedures using the arm artery rather than the artery in the leg, and that reduces the bleeding at the site. So just want to comment that, to sum up, about 5 million patients have been treated with Abciximab worldwide since its approval, and another 5 or so million patients have been treated with one of the other two drugs. And at 6 months, the reduction in the likelihood of either having a heart attack or dying from the first heart attack is reduced by about 2.6%. And so that translates into about 260,000 people. This is a political year, so I thought I would show you how we've evolved the therapy of heart attack over the last half a century, or more than 70 years, close to now. I remember when Eisenhower had his heart attack. It was a major event. The stock market crashed. I think would be well over 1,000-point drop in the Dow today. His doctor was with him, but he didn't even do a cardiogram till the next day. He did give him a shot of morphine, and they did have anticoagulants back then and drugs for irregular heart rhythm. But the real treatment at that time was 6 weeks of bed rest, and starting with absolute bed rest. In fact, they sat him up in the first week because that was a novel thought that people had. They didn't like what happened when they sat him up, so he actually had 7 weeks of bed rest. And somewhere between 30 and 40% of people died. You contrast this to 2000 when Vice President-elect Cheney had a heart attack, and he was in the angioplasty suite within a couple of hours. He got these drugs and probably got these drugs as well. He walked out of the hospital in 2 days, and now it's more like 1 in 20 rather than 1 in 3 dying. And in the last year, you may remember that Bernie Sanders had a heart attack, and he was back on the campaign trail within a week, and people stopped talking about this as a major issue, which is sort of amazing given what has been in the past when people had heart attacks. So where is the real need now? Well, as Rick mentioned, a lot of people die before they get to the hospital, but more important, even if they don't die, they may lose a lot of muscle before they get to the hospital. And you can see these data from Sweden. This is taken from patients 65 to 74 years of age. And you can see that just like in our country, there's been a dramatic drop in how people, the improvement in the survival of people of having a heart attack while they're in the hospital. But you can see from the green that there's been much less improvement in the people who die from their heart attack out of the hospital before they get to the hospital. And there are a lot of reasons for why we have not been able to do better. Where people have really tried very hard, they can show benefit. So this is a study in which they gave Abciximab in the ambulance or at the time of the angioplasty and the stent placement. And you can see here when they hadn't given Abciximab before, only 7% of the patients had what I'll call an open artery, and that's called TIMI-3 blood flow. And about 12% had partially opened artery, and that's called TIMI-2. TIMI-0 is no flow, and TIMI-1 is just a trickle, so they combined them here. Over here, they heroically got Abciximab into patients about an hour after the onset of their symptoms by giving it in the ambulance. And you can see that instead of 7% having an open artery, it was now 28%. And instead of 12% having a partially open artery, it was now 23%. And that's really the opportunity that we have. But it's not just about dying in the first couple of weeks or even the first 6 months. These are data where they pooled a lot of studies to look at what happens if you give the alpha-2b beta-3 antagonist, any one of them, less than 65 minutes after the onset of symptoms, or you can see these other time points up here, more or less at about 3 hours. And over here, this follow-up is in years. So this is 11 years later. And what you see is not only is there a benefit early on, immediately, but the actual subsequent slope of death is very different. And that, I believe, is probably a reflection of the fact that these individuals lost a lot more of their muscle as a result of not getting treated early. And as a result, they were more likely to have heart failure in the out years. And you can see this rather dramatic difference in mortality. So now I'm going to go into a little bit of the science of how we thought about trying to make a better drug. This is that receptor, but we now have a crystal structure of it, so we can really look at it in a lot of detail. And what we know is that where the fibrinogen binds is up in this area over here, and it ordinarily is in this bent form. When it gets activated, it goes into this form and this form. And these two modifications are called extension of the receptor And it's over here, it's called swing out because this region over here literally swings out. Ironically, the fibrinogen molecule, in fact, results in this swing out motion. But because eptifibatide and tirofiban, they both bind to the receptor more or less the way fibrinogen does, right in the little pocket. And as a result, they themselves will cause the receptor to undergo this major conformational change. And in addition, they put the receptor into a high affinity state so that if they come out, then in fact, the receptor will be activated for a period of time. And that is of concern. If you want to see what that swing out looks like, this from a study we did, Tim Springer did the crystallography, where this is showing what happens when you have one of these small molecule, eptifibatide or tirofiban, come in, and you see this major shift in the conformation. There's another reason this conformation may not be so good, and that is about 0.5% to 1% or 2% of patients develop low platelet count when they get one of these drugs. And the current hypothesis is that regions of the receptor that ordinarily are not exposed, that only get exposed when the drug comes in, are sites that some people just happen to have floating in their blood antibodies that recognize these sites. Because if you can show that when you react their antibodies with normal platelets, they don't react. But if you react them with normal platelets in the presence of the drug, they do react. So what would be an ideal antiplatelet agent for early therapy? Well, we'd like it to be potent because the more potent it is, the more likely it is to work. We'd prefer that it doesn't change that shape in any way, that paradoxically activates the receptor and may result in a low platelet count. And it would be good if we didn't have to give it intravenously. All of the agents that I mentioned need all of the agents that block this receptor need to be given intravenously at current, but that's really hard to do in an emergency situation. And just giving it directly under the skin, it would be great if it had rapid onset because that's what we really care about. And rather than having a long duration, we'd actually like it to have a short duration to limit the risk of hemorrhage and to let the doctors in the hospital decide, for example, if a patient needed to go to surgery. And it has to be soluble enough. That means you have to be able to dissolve enough of it in just under 2 mLs in order for it to be administered by an autoinjector, because realistically, that's about the maximum that you can get. I want to just come back to this slide for 1 minute and just show you this little magnesium ion that's in this receptor, because the magnesium ion is really crucial for the binding of fibrinogen and for these other agents. And that's really the key to what we were interested in thinking about in terms of making a new drug. So after I came to Rockefeller, I learned that we had this great high throughput resource center where we could look at as many at that time as 33,000 compounds. And so what we wanted to do was to put fibrinogen on a plate, put in platelets, and see if any of these 33,000 compounds would block the binding of the platelets to the fibrinogen. This was relatively new technology back then, and it was great that we had access to this at Rockefeller. Rockefeller was really at the front of developing this technology. Right now, I'll just tell you we have over 300,000 compounds to give you some idea of where things have gone. And amazingly enough, we found one out of those 33,000 that looked interesting. And we called that, actually, I call them RUC1. If you want to break the code, it's not very complex. It's Rockefeller University Compound 1. At any rate, you can see that if you add more and more of it, you can inhibit the platelet aggregation that I showed you. And if you give it to a mouse, you can prevent the mouse's carotid artery that I showed you from closing. Here's the control mouse, and you can see that within 12 minutes, almost all of the mice have a closed artery, but RUC1 prevents that. But RUC1 really wasn't very potent, but it gave us at least a clue. And that allowed us to move on and design RUC2. And RUC2 was about 100 times more potent and pretty close to Eptifibatide. And we were then able to do crystal structure with Tim Springer's group back up at Harvard. And what we could show was that RUC2 was really interesting because what it did was, rather than binding to that magnesium that I showed you, it kicks the magnesium out, which means that fibrinogen can't bind because there's no magnesium for it to bind to, which means that there's nothing to come in and cause the conformational change that I showed you. So RUC4 actually locks the receptor in the inactive conformation as opposed to actually activating it. But RUC2 didn't, so I'm back to RUC2, I'm sorry, I'm jumping ahead here, but RUC2 had one lethal problem. Remember I said that it has to be soluble enough to get into an auto-injector. And for whatever reason, it wasn't, even though chemically it looked like it probably should be. But we thought maybe the reason that it wasn't was that it was self-associating on this ring. And so we interrupted that, and you can see the dramatic increase in the solubility. And this then met our criteria. And here you can see that the IC50, the amount of the drug that we needed to inhibit the platelets, actually went down, which is good, because that means you need less drug to inhibit it, means the drug's more potent. And this RUC4 then became the agent that we began to work with, and it also was able to protect mice from the occlusion in their carotid artery. And when we gave this to animals, you can see that when we tested their ADP aggregation at the beginning, it was quite fine. But within 15 minutes of giving it intramuscularly, in this case, you can see that there's no aggregation. And this lack of aggregation continues on for several hours and then comes back. And this is a model in a mouse that is designed so that human platelets interact when you damage the vasculature. And these are fluorescent human platelets then. So we're now looking at no drug or RUC4 or Abciximab to see what they do in this model. And over here, you can see that if you don't have the drug at all, the platelets heap up one on top of each other. Whereas with RUC4 or with Abciximab, they just don't do that because they really can't recruit additional platelets easily. They will adhere because they have those other receptors, but they just can't build up, and many of the platelets just break off and go downstream. So there's a bit of a major period of time between doing those studies and actually getting the first human studies. But this was done with Dean Kariakis's group out in Cincinnati. And this was started with healthy volunteers who were eager to participate in this study. And here you can see that when we do the aggregation studies, that we're getting about 70 to 80% inhibition within 15 minutes at these two doses. And the reality is that we know a lot about these drugs, and we know that we really want to be a little bit above 80% to reach the maximum antithrombotic effect. The next set of studies were done in patients who had stable coronary artery disease. So they weren't having a heart attack. They were just they just happened to be on aspirin for their heart disease, but they were feeling well. And again, we used the same two doses. And in this case, again, we got about 70, 80%. And you can see that this comes back over a period of about 2 hours to the baseline value. And then we finally got to begin to treat patients who are having a heart attack. And there's something, just this abbreviation, this ST segment elevation myocardial infarction. This is a particularly well-described type of heart attack in which patients, you almost always have a clot as the basis for it, and we wanted to focus on that. These patients were on aspirin, heparin, and another antiplatelet agent. And here you can see that at somewhat higher doses, we're now consistently getting results above here. In this case, we used another assay. This is actually an assay that I worked with the company called Accurmetrics to develop, and it's automated, so it's easier to do, and it correlates well with the other assays. And now we're getting ready for studying a study with over 1,600 patients. And here they'll be given either a placebo or 2 different doses of RUC4 in an ambulance. And this is one of the kits that will be in the ambulance where we would have started a while back if it weren't for COVID, but we're just about ready to start now. And some of these kits will contain placebo, and some of these kits will contain the active drug. So if you right now arrive at a center that can do this procedure, on average around the country, it's about 67 minutes before you actually get the procedure. So giving RUC4 in the emergency room would potentially provide protection through that period. Giving it by EMS during the ambulance would provide additional protection for the loss of muscle along the way. And of course, ultimately, if this proves safe enough, and that will be the key issue, we know that there will be the possibility, at least, for self-administration. So here's among our dreams, that here's the drone that has in it something that can hook up to your smartphone to send your cardiogram directly to your doctor. And if the decision is made that you're having the heart attack, then maybe we can send you the drug in the same drone. Now, there are other things that we dream about, and I guess that's the right word. But as I mentioned, we know that myocardial infarction in countries with limited access to mechanical coronary artery interventions and stent insertion is a real problem and a growing problem. So we hope maybe we can make a contribution to that. People who have out of hospital cardiac arrest and get treated with an automated external defibrillator and get their cardiac rhythm back, a substantial fraction of them will have the kind of pattern on their cardiogram that suggests that they may be having a heart attack, or that the heart attack is really what triggered their sudden death. And so if it turns out that we can actually improve the therapy of those individuals by treating them at that point, that would be great. And there are 16 million Americans who've already had a heart attack. And if they obviously are at high risk of having another heart attack, and that potentially could be an opportunity for very careful introduction of self-administration. So it's really crucial that I reiterate what Rick pointed out before. We really live in an enriched environment at Rockefeller. In many ways, I'd say a uniquely enriched environment. And as I've already told you, the high throughput screening facility was absolutely crucial for us doing this. We also had great help from the Proteomics Resource Center because we had to set up an assay when we were doing the RUC2 studies to actually measure the amount of RUC2 in the blood of the animals we were studying. None of this really would have gotten past animal studies without the Robertson Therapeutic Development Fund, because the monies that they gave us really made it possible to move to phase 1. And I just really want to emphasize the heroic and courageous people in the hospital and in the Center for Clinical and Translational Science. We use a lot of human blood in order to develop these drugs. And also the design of the early studies were done with the broad array of experts in human subjects protection, clinical trial design, the regulatory issues, the bioethical issues. All of these are profound, and we are so fortunate to have an infrastructure of people with that expertise. And the Office of Technology Transfer played a crucial role in the licensing of RUC4 to a new company called Cellacor. For those who are interested in timelines, I think this gives you a little taste of what drug development really is like. So the screening of the compounds, we published our first paper in 2008, and actually the screening started before that. We got to RUC2 about 2012. That's when we published it. We published about RUC4 in 2014. The Robertson grant was in 2016, and then we had to work our way through the FDA and submit an investigational new drug exemption. And then the phase 1 that I mentioned, the phase 2A that I mentioned, and now we're about to start the phase 2B. And the last slides just give me a chance to recognize so many people who helped us so much around the institution and around other institutions, our medicinal chemistry colleagues, our crystallography colleagues, our computational colleagues, colleagues with the animal experiments, our pharmacokinetics and our animal studies. It really is, and now, of course, many people at the company Cellacor. Thank you for your attention.
So Barry, that was really fascinating. We have a number of questions that have come in. One of them is from a high school student who asked, how did you land on this target to focus on for your research to begin with?
That's great. That's the perfect question, and I didn't have time to go into it. We learned about this target because of patients, and Dr. Lifton himself is the world expert on using interesting, challenging patients to learn about the fundamentals of biology and using the genetics to help us understand human physiology. And so there is a group of patients who have a disorder in which they either lack this receptor or the receptor doesn't work properly. They go by the name of Glanzmann thrombocythemia. And we have studied those patients for many years, including doing prenatal diagnosis and identifying the genetic basis of their disorder. But they have a lifelong bleeding disorder. The most important thing is to really study patients very, very carefully, because one of the things that was obvious was even though they had a lifelong bleeding disorder, fortunately, they didn't develop spontaneous bleeding into their brain very commonly. In fact, it was very unusual unless they had trauma to their head. And as a result, that's what let us hope that maybe we could get away with essentially blocking this receptor for a finite period of time without an unacceptable risk of bleeding.
Terrific. So another question has come in. Until we have this drug available for drop-off by Amazon at the onset of symptoms, what should patients do if they suspect they're having a heart attack at home before the ambulance arrives?
Sure. The first thing is to recognize and not ignore the symptoms. And I'm sure that Holly Anderson would like me to emphasize here that the classic symptoms are not as common in women. And so one needs to be really attuned to what those symptoms are, in addition to the classic chest pain, and people have talked about it feeling like an elephant just stood on their chest, and pain running down your left arm. But much more subtle symptoms we know can be associated with having a heart attack, including things like shortness of breath. So I would say the most important thing is to overcome denial because time is of the essence. The issue of whether one self-medicates with aspirin, if there's Certainly, if one was in a high-risk group, one would presumably already be on aspirin. But if one was in a high-risk group and had presumptive symptoms, of course, you call your doctor first. But my suspicion is your doctor would probably say aspirin is a reasonable thing to do. We know from early studies that decreases the risk of mortality by about 25%, just that one simple act. So the most important thing, though, is to get that artery open as quickly as possible.
Great. So how are you thinking about the mechanism of action of RUC4 in terms of, are you busting open the thrombus that's sitting in the coronary artery, or are you simply preventing propagation of the clot? What do you think the goal is?
So that's also a terrific question. So people, and I'm sure I'm one of the culprits here. And when we present this, we sort of make it sound as if that clot is an instantaneous final event, as if it all happens in the first second. And that's not really what happens. And in fact, that's one of the reasons I showed you these videos of the damage, because you can see that it takes a while and that these are quite dynamic early on. The other thing is, the earlier the clot is, the more likely it is to still be rich in platelets and still be reversible. So that's another reason why an antiplatelet agent is more likely to work the sooner you can get it into someone, because it's still likely to be a dynamic thrombus, which is sitting down and breaking off. And if you can then at that point prevent new platelets from getting into that thrombus, in short order, you may well have an opening of that artery. We do have our own endogenous thrombolytic system that generates an enzyme called plasmin that can actually digest fibrin. So we do have our own fibrinolytic system, that's what it's called. And you may remember that it used to be more popular to give tissue plasminogen activator, a clot buster. But again, the earlier, the better, because the more dynamic and the more platelet rich.
So another question that's come in is, how would RUC4 interact with other drugs that you might be administered in the immediate, in the minutes after onset of heart attack, and if you're headed to the cath lab, for example?
Sure. Well, this is very challenging because the standard of care keeps changing. In the United States, if an ambulance comes for you, they'll probably give you an aspirin, but they probably will not give you any other drugs if they think you're having a heart attack. Although some may be giving one or more of those drugs against that ADP receptor. Remember, that's the class of drugs that includes Plavix. The problem with those drugs is that they're all oral drugs. And when you're having a heart attack, you don't absorb oral drugs well. All of those drugs have been studied to be given in early on, like in an ambulance, but they've been very disappointing and have not been able to show significant improvement. But when you actually measure the antiplatelet effect usually takes several hours to reach its maximal. And so those oral drugs really just can't work very well. But just say that in Europe, if the ambulance picks you up and they think you're having a heart attack, you will not only get aspirin, but you will get one of those drugs that's against that ADP receptor, and you'll also get heparin. So there's enormous variability in what is considered standard of care. And virtually no places are doing the heroics of trying to get Abciximab or those other drugs that have to be given intravenously into people pre-hospital.
Continuing on that question about the controversy of what standard of care should be, for years people have been told that if they've had a heart attack or otherwise at high risk to take a baby aspirin. This has been questioned recently. What are your views and thoughts on the role of low-dose aspirin therapy in patients at high risk?
Yeah, so I would say if you're having a heart attack, that's not a place for low-dose aspirin. In fact, in Europe, they give intravenous aspirin to make sure that it gets in because of the same issue of absorption from oral agents. The other thing is that you really, some people will encourage you to chew the aspirin because that'll increase the speed of its absorption. So when you're having a heart attack, a low-dose aspirin is not the preferred.
How about in the chronic setting of just prevention?
Okay, so we get into a very, very complex set of questions now. And I think that to distill this down, I actually spoke about this soon after I arrived at Rockefeller. So if you have had a heart attack, I don't think anybody would disagree that you should be on aspirin thereafter. If you haven't had a heart attack, we're now into an area that we call primary prevention, meaning you haven't had any evidence of that. And at that point, most people will take a look at your 10-year risk, or some people think it should be a lifetime risk, because there's no question that aspirin is not a perfectly benign drug. And so we would restrict its recommendation to people who are at high risk. Exactly how high risk, that's a controversial thing. So studies where the more recent studies have not really shown much, if any, benefit in people who are at low risk for having a heart attack, being on aspirin, and it may confer real risks. Having said that, there's also an equally interesting controversy about whether there's a role for aspirin in preventing cancers, particularly gastrointestinal cancers. And to be honest, I think that's the subject of a whole other hour discussion.
So in your clinical trial, as it moves forward, what are the critical readouts going to be?
Perfect question. So we want to know if the drug is working the way we think it should be working. And as a result, the number one thing we're going to look at is how many arteries are open when the patient actually gets to the angioplasty suite. Because like in that experiment, that study that I showed you, we would anticipate that if this drug is doing what we think it's doing, more patients should get there with an open artery than with a closed artery. So that's number one. But we have joined that with whether or not we think this is going to actually have an impact. And so an hour after they have the procedure, remember I mentioned that STEMI stands for ST segment elevation, myocardial infarction. Well, it turns out the speed at which those elevated ST segments, that's a part of the cardiogram, the speed with which those return to normal is a very, very good indicator of what's going to happen to you in the next year, whether you're going to die, whether you're going to have heart failure, whether you're going to have other problems. And so 1 hour after the procedure, we'll be looking to see what is the impact on ST segment elevation, the return of ST segment elevation, how close it is back to where it should be.
You emphasized the long timelines for drug development. So if all goes well, when can we look for RUC4 to become available?
That's also, of course, a terrific question. And it will depend on a lot of things. It will depend on how our phase 2 study goes. It will also depend a lot on whether or not we can raise the money for a phase 3 study. Phase 3 studies that will meet the regulatory requirements of the FDA for a drug like this, not uncommonly will cost in excess of $100 million. So that's a big bet that somebody has to be willing to make in anticipation of a reasonable economic return. And of course, for us, it's the big bet in terms of whether or not we can really have an impact on people worldwide.
Do you think another question that's come forward is How hard is it to standardize activity, for example, in an ambulance? If you had an effective therapy, trying to get everybody to use the drug the same way seems like a very challenging logistical problem.
It is, and I could share with you the training program that has gone into developing the phase 2B study. It's a very, very elaborate training program for the personnel. We've done a lot of focus groups with EMS personnel. We've designed things and redesigned them in response to the preferences that we've heard from the individuals who were going to be in the ambulances. And we've redesigned the way in which the dose is calculated, the way the syringe is drawn up. And training is absolutely the most crucial aspect of maintaining high quality discipline. And then auditing and monitoring. We're lucky at Rockefeller. We have great auditors and monitors for the studies that we do in our hospital. And as a result, we have the ability to make mid-course corrections. And so it's not like you just start on day one and wait till the end. You're constantly overseeing and identifying whether or not there are any things that need to be addressed because you just couldn't possibly anticipate everything.
Well, Barry, I want to thank you for a beautiful presentation about your incredibly exciting research, as well as for your years of leadership here at Rockefeller. It's time to close our discussion now, and thanks for all of the great answers to questions, and thanks to everyone in the audience who participated by sending in your questions. But it's been a fascinating hour, and thank you so much for everyone's participation. I want to thank everyone for joining us, and I'd also like to thank you all again for your generous support of the university. All of us here at Rockefeller hope that you will stay safe and be healthy. Thank you very much for participating this afternoon. Good evening.